Good morning, biology, and welcome to chapter 3. In this chapter, we're going to be talking about biochemistry, or specifically looking at the science of life processes. Last chapter, we talked about the chemistry of biology, and we talked a lot about the kind of non-living components of chemistry that are really important for biological processes. Things like covalent and ionic bonding, things like hydrogen bonding, the properties of water, all this really good background information that allows us to jump into biochemistry, where we're taking bio, which is the study of life, and chemistry, chemical functions, and putting them together, and talking about how living things use biochemistry to be alive. All right, so the first thing we need to talk about is there are two types of compounds we're going to talk about. There's inorganic versus organic. And organic are those that have carbon that are found in living objects. These are things like DNA and sugar and ethanol even is a organic substance that is created by yeast, methane by cows, and lots of other bacteria and other organisms. These things are primarily made up of carbon. They have long carbon backbones. They have lots of carbon in their molecular structure. Anytime that you eat sugars or steak or anything like that, you're eating lots of carbon. Inorganic compounds are things that do not contain carbon. Things like table salt, which is NaCl. Things like hydrochloric acid, which is HCl. Things like quartz, which is mostly silicon oxygen. The one kind of inorganic compound that has carbon that's important to note is carbon dioxide, CO2. While it has carbon in it, CO2 is not an organic compound. We breathe out carbon dioxide as waste, so it is an inorganic compound. All right, so we need to talk about the element of carbon. We need to talk about why carbon is important for the biochemistry of the body of all living organisms. Carbon has 4 valence electrons, these electrons that are on the outside edge of it. It wants to get 8 electrons on the outside here, so it wants to form 4 bonds. One here, one here, one here, and one here. And unlike our other elements, it will form long chains with itself that are longer than 2 or longer than 8 chains of these elements. This will form chains, rings, and branch chains of carbon molecules that we'll talk about coming up. Each of these has their own unique chemistry and allows the body to do unique things. So the first thing we need to talk about is bonding. And one bond between 2 carbon atoms or between any 2 elements is called a single bond. And in this, only one pair of electrons is being shared. It is important to note, however, here that there are 2 electrons being shared here, one from each carbon atom. They're making this bond, so this carbon has partial ownership of these 2, and this carbon has partial ownership of these 2 as well. This is what creates this bond. In a single bond, they're just sharing 2 electrons. As we get a little more complex and we move into double bonds, now we're sharing 2 pairs of electrons. So for each one of these little guys here, there is 2 electrons in that one and 2 electrons in this one. So how many individual electrons are being shared here? There are 4 individual electrons being shared in this double bond, where each of these carbons has partial ownership and kind of sharing the ownership of these. Double bonds are going to be shorter than single bonds, and they're a little bit longer than triple bonds. In a triple bond, this is when carbon is doing 3 pairs of electrons. So once again, how many individual electrons? Well, there's 2 in this one here. There's 2 in this line here. And there's 2 in this third line here, for a total of 6 electrons being shared there. This carbon and this hydrogen, each one of these are sharing an electron in a single bond, and this carbon and this hydrogen. So what this ends up looking like is each one of these carbons has 8 total electrons, which is that number that it wants to have. Where each one of these has 8 total electrons that it's sharing, making it a full happy element. All right, functional groups. These are groups of molecules that are attached to carbons, usually, or to other elements, that give them a new characteristic or a new trait. An example is this OH. This is when a hydrogen and an oxygen are binded together. And we call this group hydroxyl. And this makes whatever molecule they attach to polar. So if they attach to this carbon here, we have a bunch of H's coming off this carbon, this is now methanol, which can be used in hand sanitizers and other kind of things. This one is a polar molecule. Remember that it's hydrophilic. It loves water. So this means that this would be dissolvable in water, this molecule here, because it has this OH group. This OH group here is polar and makes it so that it would dissolve inside of water. All right, some other important functional groups. We talked about hydroxyls here. This one's on ethanol. Carboxyls are this C that's double bonded to an oxygen and then single bonded to a hydroxyl group here. There's amino. This is where N is bonded to an H and H. There's no carbon here, but it can bond to carbon in molecules. And then phosphates are really important. These P that has these 4 oxygens, one of them double bonded. These are really important for our body's energy systems and your DNA structure. Carbon likes to bond together to form large carbon molecules. But the simplest, smallest piece is called a monomer. And a monomer is a small, simple molecule. This here is an example of a monomer. They bond together to form polymers. So this polymer here is made of one monomer here, one monomer here, one monomer here, and one monomer here, for a total of 4 monomers in this polymer here. So these polymers use these monomers to make long chains. And when we get these larger polymers, we call them macromolecules. And the word macro means big versus micro, which means small. So macromolecules are big molecules. So every time we add a monomer to make these kind of large polymers, we need to do what we call a condensation reaction. And we call it a condensation reaction because we're losing water. So in this example here, we have glucose, which is a type of sugar, and fructose, which is another type of sugar. They're going to bond together in what we call this glycosidic bond here. But when they do, this OH group and this H over here are going to leave and combine to form H2O. So condensation reactions, water is released in these. They are the opposite of hydrolysis reactions. Hydrolysis reaction. The breakdown of polymers, so to build them, we use dehydration synthesis or condensation reactions. To break them apart, we use something called hydrolysis. And like before, we're going to split this word into 2 parts. The hydro part means water, and the lysis part means cutting. So hydrolysis means water cutting. And what we do is we use water, as you can see up in this diagram here, we use water to come in here and it'll attack this bond and it'll split these into these smaller pieces. So this is what your body does when it wants to break down substances into smaller parts that you're able to use. Now we've talked a lot about energy and the body requiring energy, but we need to talk about what our body uses for energy. So life processes require a constant source of energy. Your brain needs blood sugar at all time. If you, for some reason, don't have blood sugar in the brain, you've got about 6 minutes to get blood sugar to the brain, or you have permanent brain damage and then usually death shortly after. To store energy in the body, we use this molecule here called ATP, or adenosine triphosphate. And it's called adenosine triphosphate because it has an adenosine here, which is a nucleotide, and then it has a triphosphate, or 3 phosphates. 1, 2, and 3. These phosphate bonds, especially the one on the end there, is really easy to break off, and this phosphate can be used to produce a lot of energy. When we talk about glucose and glycolysis and photosynthesis, this ATP becomes very, very important, and we will use it a lot. Most of your body's processes require ATP in some form. This is the conclusion of 3.1, but we're going to jump on in and start 3.2 in this video as well. And in 3.2, the kind of topic that we're going to go over is biological molecules. So we've talked about monomers and polymers, but we're going to talk about an inorganic versus organic compounds. But now we need to talk about the different classes of these organic compounds. So all of these are going to use carbon. So that means that they are organic compounds. But the four big classes of biological molecules are carbohydrates, lipids, proteins, and nucleic acids. The first, carbohydrates, these are your sugars and starches. And everything in this picture contains a lot of carbohydrates. These grapes contain a lot of carbohydrates in the form of sugar, a lot of starches in these rice and grains and breads, all of these, the sweet potatoes. And carbohydrates contain lots of hydrogen, oxygen, and carbon. And the H and the O, the hydrogen and the oxygen, will always appear at the same ratio as water. Water is H2O. Carbohydrates will always have that 2 hydrogen to 1 oxygen ratio. So if you have some carbohydrate that has 7 oxygen, it's going to have 14 hydrogen. And we classify these carbohydrates based on their size. There is one really important carbohydrate you need to know. And this really important carbohydrate you need to know is glucose, which its chemical structure is C6H12O6. And you need to memorize that. That is an important, that is the one carbohydrate that is really, really important for this level of biology. This is what glucose looks like. It has a ring structure. It's a hexose sugar, meaning it has 1, 2, 3, 4, 5, 6 carbons. What you need to know is C6H12O6. And that's the same glucose that's in plants, the same glucose that's in humans, the same glucose that's in fungus. All has that C6H12O6 structure. All right, so for our carbohydrates, we're basing them on size. The first ranking here is monosaccharide. And mono means one, and saccharide means sugar. So this means one sugar. These are our simple sugars. They either have a single chain or a single ring structure. They're not bonded to other sugars. Glucose is the most common of these. We call glucose blood sugar. Two other important examples are ribose and deoxyribose. These sugars will be used in our nucleic acids, in our DNA and RNA. But any monosaccharide is just one single ring or chain structure. Some other important monosaccharides is fructose, which is found in fruits, and galactose, which can be found in milk. Our next, and moving up in size, is disaccharides. And di means two or double, and saccharide means sugars. These are our double sugars. These form when our two monosaccharides are joined through a dehydration synthesis. So they're losing H2O. H2O is escaping into the environment. And these guys are now bonded together. This is not good if you are trying to use it for energy, but this is really good if you're trying to use it for storage. So disaccharides are too large to pass through cell membranes. So this means that if you get it inside of your cells, you can use it to store it there because they can't get back out. If you're ingesting it and you're taking it into your body and you want to use it and get it into your cells, you have to digest it down to monosaccharides, down to those simple sugars. To do this, we use hydrolysis to break apart that bond, which will release our simple sugars that can then be digested. In this example here, this water is being used to break apart sucrose using the enzyme sucrase to create glucose and fructose, which your body can then use to make energy and do cellular processes. Our next one is the polysaccharides. And poly means many, and saccharide, of course, means sugars. These are many sugars. There are many sugar molecules put together. They have long, branching chains of linked simple sugars. This starch here, this amylopectin, is an example. This is found in sweet potatoes. And this is really, really good for storing a lot of sugars in a kind of space. They lack sweetness, however, because of their size. So even though you can eat a potato and you're getting lots of sugar from it, you don't taste that sweetness. In order to taste it, these sugars have to be really simple and able to dissolve on your tongue, and starch is not able to dissolve on your tongue. So that's why you don't get a sweet taste from starchy foods. All right, next up we're going to talk about isomers. Isomers are chemicals with the same chemical formula, but they have different structures. So there's things like glucose, mannose, and galactose here all are C6H12O6, but how they are kind of oriented is a little bit different between them. The difference between glucose and mannose here is this OH group is swapped. It's on the different sides there. Same thing again, this is swapped here. But then this OH group in galactose is swapped as this group in glucose. So even though these guys are all C6H12O6, how those elements are oriented in these molecules is a little bit different. And that gives these guys a little bit different properties and characteristics. All right, so on this slide, we're going to talk about 3 important polysaccharides or many sugars that you need to know. The first is starch, and this is a storage polysaccharide used by plants. It has this long branching structure here. This is what is used in potatoes and carrots. It allows them to store a lot of energy inside of them. Glycogen is a very similar to starch, but is smaller, and it is found in animals. And it is formed by linking just glucose chains together. And glycogen has these big long chains here. These chains are shorter, they branch more often, but they're really good for storing glucose. This is really common in your liver cells. When you start exercising and you start burning muscle and fat, this is what gets broken down first to replenish your supply. And then our third polysaccharide is cellulose, which we call fiber. And this is what gives plants and plant walls their solid structure. Cellulose is not digestible by humans. This is why you don't get any benefit out of eating bark, but some organisms, such as pine beetles and other organisms like that, have bacteria that allow them to break down the cellulose and use it for energy. The nice thing about cellulose, and it's strong, it's packed into these really, really tight rows, is it gives you a lot of strength and rigidity to wood and allows trees to stand really tall against the elements. Next up, we're going to talk about proteins. And proteins have carbon, hydrogen, and oxygen, just like our carbohydrates, but they also contain nitrogen and some contain sulfur. And each protein is made of individual units or monomers called amino acids. And this here are all things that you're getting proteins from. Things like broccoli, things like meat and eggs, cheese, oats have some in it, spinach. Spinach doesn't have as much protein in it as you'd think it would. Popeye, that he ate spinach and he was all big and strong. That was a misconception. It was a misprint of how much protein was in the spinach. But proteins can be found in these foods, many others, and they're very, very important for the survival of life as a whole. All right, as we said, all protein is made up of amino acid. There are 20 different amino acids that most living organisms use, and they all have a very similar structure. They have an amino group, which is this nitrogen and 2 hydrogens. It gives them basic properties, or pH above 7. And they have an acid group, or this carboxyl group. This carbon double bonded to an oxygen and an oxygen and an H. This gives them acidic properties. So these guys have both basic and acidic properties, and this gives them a lot of chemistry that they are able to do. Where they differ is they all have different side chains. We call these side chains R group, and they give them very different structures and very different functions to whatever protein they're on. These are the 20 different amino acids. They all look pretty different. Some of them look a little bit closer to others. But this is what your body uses to make all the proteins in your body, is these 20 amino acids. When 2 amino acids come together, like we've made a disaccharide, we can make a dipeptide. And this dipeptide shares a bond called a peptide bond. This peptide bond is in between the carboxyl group of one and the amine group of another. And since this is a synthesis reaction where we're putting two things together to make something new, we're losing a water in a dehydration synthesis, just the same way as we were in with the carbohydrates. All right, next up, we're going to talk about polypeptides. And these are long chains of amino acid that are all linked through these peptide bonds. So in this one here, there's about 30 or so of these guys all bonded together. All these different three-letter abbreviations are different amino acids. And if a whole bunch of these chains are put together, they can form a protein. Hemoglobin, the molecule that carries oxygen throughout your body, is made of 4 chains of protein. It's made out of 4 polypeptides. Also, important to protein chemistry and biology are enzymes. And enzymes are protein molecules that are acting as catalysts. And most of the enzymes in your body are proteins. There are a couple of them that are nucleic acids. We'll talk about those just briefly. But enzymes work on this very unique fit that they have between the enzyme and whatever substrate they're working on, or the molecule they're working on. This molecule that fits into the enzyme's active site here is the substrate and is the one that is being catalyzed. This is going to change somehow. On an enzyme, we have something called the active site. And this is the shape that allows the substrate, a specific substrate, to fit in there. When this happens, it changes the enzyme shape. And now that this enzyme shape has changed, the bonds of the substrate that we're trying to change weaken, and the activation energy for the reaction is lowered. Without these guys, the reactions in your body would take about 1 million years to complete naturally. And in your body, they happen at 100 times a second, 1,000 times a second, very, very quick rates. The thing to note about catalysts is they don't change. While the substrate is coming in and being changed, the catalyst itself doesn't. So after the reaction, after these two molecules are coming in here and getting bonded together, they are released as a product, and this enzyme reconfigures itself and is ready to do another round of this. And it can continue to go on and on and on until there's no more substrate available. Enzymes are really picky, though, about two things. They're picky about temperature, and they're picky about the pH. So if either temperature or pH changes, usually these enzymes aren't very good catalysts, and they're not as effective. All right, our third class of biomolecules here are the lipids. These are very large, non-polar organic molecules, meaning they don't dissolve in water. They include things like triglycerides, phospholipids, steroids, waxes, and pigments. These food groups here are the ones that you'll find lots of fats in. This is fish, olives, any kind of nut or oil. Avocado has lots of them. There's sunflower seeds here, flax. All these good things have lots of lipids in them. Another place we see lipids is on the outside of this leaf here. This cuticle, we call it, on the outside of the leaf here, is very waxy structure. And this wax prevents this water from standing on top of this leaf. It causes it to beat up and run off. This prevents the plant from getting a lot of mold infections that it would normally get, or substances that are wet would normally get. So these substances help a lot in biological organisms. The composition of lipids. They have a lot of carbon-hydrogen bonds. Every single one of these lines here is carbon molecules that are bonded together with lots of hydrogen around them. In a single free fatty acid here, we only have 2 oxygen. So there's not as many oxygen inside of lipids. If you see a molecule that has mostly carbons and hydrogens, something like let's go C24, H48, O2 would be an example of a sample lipid molecular structure. There's not as many oxygen as in our carbohydrates or even in our proteins. The function of lipids is to store energy really, really well. And in fact, they are the best at storing energy. They store more energy per gram than the rest of organic molecules. When your body tucks away energy in fat, this is what it's being tucked away as, is in lipids. All right, so we're going to talk about the several different types of lipids. First off, we have the fatty acids. These are unbranched carbon chains. One group has the CH tail out here, and the other group has a polar head. This polar head is usually a carboxyl group, where it's that double oxygen bonded to an OH molecule. The polar head is hydrophilic. It loves water. It wants to be near H2O. The other end, however, does not want to be near H2O. It wants to be near more carbons and more hydrogens. So there are two types of fatty acids. There are saturated fatty acids and there are unsaturated fatty acids. And you might have heard that unsaturated fatty acids are good fatty acids and saturated fatty acids are bad fatty acids. It's partially true. A diet that's super high in saturated fats can end up in causing some arterial blockage. But like anything, as long as it's in moderation, saturated fats aren't bad by themselves. Now, animal fats tend to be saturated, and what this means is that all the carbons in this saturated fat here have a single bond. Every single one of these carbons is only bonded to another carbon by one bond. In an unsaturated fat, which is really common in plants, these carbons can have double bonds or triple bonds where they are bonded to another carbon twice or three times. The thing about unsaturated fatty acids is they have a higher concentrated source of energy. They are able to get into kind of some weird configurations. They're able to get really tightly close together where the saturated aren't as much. The next type of lipid are the triglycerides. These are three fatty acids, usually, and a glycerol. And glycerol is this molecule here. What happens is this glycerol gets bonded to three triglycerides. And in all the organisms we see, the glycerol usually stays the same, but the fatty acids change. So in animals, these usually tend to be solid fats. And in plants, they tend to be liquid fats. So this is why when you get like vegetable oil, it's a liquid versus when you get like bacon grease or bacon fat, which turns into a solid at room temperature. The next type of lipids are the phospholipids. These are similar to the triglycerides, but instead of three fatty acids, they contain a phosphorus group and two fatty acids. This phosphorus group on the top has a charge, giving this molecule some polarity, giving a plus and minus kind of side. These are used in cell membranes, where the phosphorus head is more closely attracted to water. It really wants to be close to H2O. And the fatty acid tails are going to move away from the water. And they're going to move so that they're all kind of facing each other, forming what we call a lipid bilayer. And lipid bilayer, this is all your cellular membranes are made of two layers of phospholipids. We call this bilayer because it's two layers, an inside layer and an outside layer. The intracellular is the inside, the extracellular is the outside. This keeps the outside water out and the inside water in. This kind of fatty acid tails here are really attracted to each other and they kind of seal this thing up like a zipper. What this allows the cell to do is it allows the cell to be selectively permeable, meaning that it gets to determine what comes in and out based on whatever the cell needs. Our fourth and final type of organic molecule are the nucleic acids. They are very important. They store and transfer important information in the cell. The chief among these is deoxyribonucleic acid, or DNA. This is found in all living organisms and contains the information that determines their characteristics. It also, as an adult and as you continue to mature, directs your cellular activity and make sure that you develop in a way that you're supposed to. So this is what determines in humans when you grow lungs as a fetus, when you go through puberty as a middle kind of teenage age, and when you reach full adulthood is all determined by your DNA, as well as your eye color, the length and kind of success of your beard coming in. All those things are coded for in DNA. We also have ribonucleic acid. This is called RNA. This is very important. We're going to talk about RNA's function in great detail later in the class, but this stores and transfers information from the DNA. In the form of messenger RNA, we'll carry it to what we call the ribosome, and the ribosome will use it to bring in these transfer RNA, and eventually this whole thing will create protein. The other kind of function we talked about with RNA is that it can work as an enzyme. We call these enzymes specifically ribozymes. If you see enzyme, if somebody says, okay, this is an enzyme we're talking about, think protein. But if they say ribozyme, think ribonucleic acid derivative. All right, and the last thing we're going to talk about are nucleotides. And these are the basic building blocks of nucleic acids. You could call these the monomer group here. And nucleotides have three components. They have a phosphate group, which is here, this phosphate group, this P attached to 4 oxygen. They have a 5-carbon sugar, whether it's deoxyribose or ribose in the form of RNA and deoxyribose in the form of DNA, and a nitrogenous base, of which there are 5 different nitrogenous bases that we will talk about later. I hope that you enjoyed this chapter. This is kind of, we're getting into the meat and potatoes of biology. I hope you know more about the difference between organic and inorganic compounds, the difference between the different organic molecules, and where do you get them from and how they're kind of used in your body. Thank you for watching, and I'll see you in the next one.